Hello guys, so welcome back again, and in this video we are going to talk about applications of PCR. PCR I have already discussed about its technique, and if you haven't seen that, I will share a link, then watch it first. Then we will talk about applications of PCR. PCR name polymerase chain reaction given by scientist Gary Mullis, who was working as a lab assistant at the Human Genome Project. So let's see first application. It is so much helpful in diagnosis of many type of disease because of its specific and sensitivity. It is highly useful for the diagnosis of this type of disorder. It can be bacterial disease, it can be genetic disease, or it can be viral disease as well. So many type of diagnosis can be done by RFLP if it some kind of mutation, because some kind of nitrogen bases adding or substitution will give you different type of length that will create some kind of polymorphism that will calling as restriction fragment length polymorphism. So now these days 98% in the cases so much helpful diagnosis technique is PCR, a real boon for such type of disorder. The prenatal diagnosis of inherited disease. So it can diagnose inherited disease by using chorionic virus sample or cells from amniocentesis. Amnion is a covering of your fetus, so you can get some fluid from there and can check or diagnosis of some genetic disease as like sickle cell anemia, beta thalassemia, or phenylketonuria. So it is also helpful in diagnosis of retroviral infection. Let's see what is it. So HIV or hepatitis is retrovirus. In this they create DNA from RNA, so it is backward position of DNA to RNA. So it is complementary DNA as calling rather than DNA. It will be a cDNA, complementary DNA. So its example is HIV infection or hepatitis infection. So it is helpful of diagnosis both type of infection. It is also helpful in diagnosis of bacterial disease as well, like mycobacterium tuberculosis and etc. So it is also helpful in diagnosis of cancer or cancer. There one characteristics you should know. There is metastasis, that is so much dangerful form of cancer, and in this cells lost their special character, we call that contact inhibition. So besides these cancer, it also can diagnose chromosomal translocation cancer, like reciprocal translocation between chromosome 14 and 18, we call that follicular lymphoma cancer. It is also helpful in sex determination of embryos. So if you grow embryos by using in vitro culture or laboratory, you can determine the sex or gender by help of PCR, but it give you some negative vibe, so it is not useful, or you can say it's not permittable in Asian countries. So this is also useful to detect sex-linked disorder in embryos. So sex-linked disorder embryo get from their parents, and it should be knowing at the right time, so you can determine of sex by the help of PCR. Next application is it is helpful in knowing DNA sequencing. So what is DNA sequencing? So DNA is made up of many single nucleotide. It is a polynucleotide of many single nucleotide. So if you anyhow know the sequence of places of nucleotide, then it will be so much helpful in genetics as well as biotechnology. So we call this technique DNA sequencing. So as the PCR technique is much simpler and quicker to amplify DNA, so it is useful for knowing DNA sequencing. So it is also helpful in gene manipulation and expression studies. Manipulation is adding of gene and cutting of gene. So if you are changing gene places, then it will give different characteristics or different proteins. So we call that expression studies. Besides, these gene manipulations are important in understanding the effect of promoter and initiator. So it is much simpler. So other application is PCR is so much helpful in comparative studies of genome. We call that phylogenetics. Phylogenetics mean you have to know what kind of relation do you have generation to generation. So we call that phylogenetics. And it has revolutionized the studies of paleontology and archaeology. PCR is so much helpful in forensic medicine. Let's see what how it is useful in forensic medicine, because at the incident, if you have anyhow a single molecule of DNA from any type of source like blood, stones, hair, semen of any individual, so you can amplify the help of PCR and identify of your criminals. So it is also helpful in forensic medicine. So it is much better in comparison with gene cloning, because it is have better efficiency and minor quantity of genetic material will be doing good. It is require minimum cost. It is requiring minimum technical skill, and nowadays automated PCR is available. You don't have to do this. All of the things will do by itself. Time factor is also the key of using PCR rather than using gene cloning. So thanks guys, it is the all applications I have talked about PCR, and if you have any problem, then you can comment down and I will try to sort it out. So thanks guys.